blind. So today what I'm going to talk about is one aspect of that very enormous project. The project that I'm leading is to get a device that we've been working on actually for about 13 years, get that into human beings within 3 or 4 years. And there are other aspects of the Bionic Eye project. One of the major research areas Riley's working on with conductive polymers, and there's another whole team of people down in Melbourne that are doing some interesting stuff with boron-doped diamond and all sorts of really interesting things. So this is just one aspect of this very large project. It's probably not very well known that the very first vision prosthesis invented was from Australia. This is a fellow by the name of Graham Tassiker in Melbourne, or Victoria rather, who invented this device. I kind of question its biocompatibility. It was a material called selenium coated on some metal, and it didn't say which metal it was. And it was placed in a human being, and she said that she could see bright light where there was nothing before. So quite remarkable back in the 50s. But we've been working on trying to get it better and better since then. So when we went to look at how you would perhaps restore vision to the blind, we could see a number of different ways that you could go about doing that. You could do chemical treatments. You could release neurotransmitters. You could do some of the spectacular work with the microfluidics and deliver very controlled doses of neurotransmitters. And you could actually get the vision system to start again. You could push on the cells mechanically. You could pump neurotransmitters in and out, or you could actually cause action potentials by mechanical disruption. You could inject electrical current, as cochlear implants have told us can restore sensory perceptions. And there are others around the world that are trying to make cells that are not normally photosensitive into photosensitive types of cells. For example, the retinal ganglion cells. If they use channel rhodopsin, you may be able to restore some or add some photosensitivity to retinal ganglion cells. Tell you that that's all functioning and all that sort of stuff. It was to tell the ARC what we want to do. So there's a cochlear implant on the right-hand side, and on the left-hand side is the device that we're building. The cochlear implant is placed behind the ear, and we're using something very similar to that to do the radio telemetry and power the device. And then the device on the left is the crux of what we're working on at the moment. We're doing a number of things with the external systems, the vision processing system, and we've learned from the cochlear implant experience that once you put in the device, and if that device is basically a terminal that you can tell it what to do, if you can change the external electronics and make that better and better and better and better, eventually you can make that device that's already implanted perform extraordinarily well. So I worked at Cochlear in the early 1990s, and people that would converse on the phone or be able to converse on the phone with a cochlear implant were quite rare, and now it's quite regular. And that's primarily due to the advances in the external processing, the speech processing strategies. So we're spending quite a lot of effort on that. And so we're doing external systems and building the circuitry to handle all that. Now this is the implantable device. This is what we're calling the wide view device. It's not a high acuity device. It's for navigation, maybe some rudimentary text-based characters, maybe some sort of obstacle recognition. It's a learning exercise, and we'll build upon this and make it better and better and better. And this is what we're calling the wide view device. So it has unit 1, we're calling it, which is behind the ear. This is a device that receives radio telemetry, rectifies that signal, sends data and power down to the unit 2, which is placed on the eye. Skip that. We're in the process of building some microelectronics for this. We have some electronics that we've been working with for quite a long time, but this is the most sophisticated one that we've tried to put together. This is a mock-up of our electrode array. I'll show you the real thing in a second. So we have this 2-wire interface to prevent corrosion. There's a charge balance signal that comes down that line. We then have a hermetically sealed capsule made of ceramic and titanium. We then have a platinum and polybimethylsiloxane or silicone rubber electrode array. Only materials that we have that have a strong history in surgical implantation, long-term implantation. We don't want to reinvent anything right now. This device has got to be in very quickly as we can possibly make it. We don't want to be introducing new materials. Now, other devices that we are working on, they will be other materials in that will have to be qualified, etc. This one's a very conservative approach with nothing new in terms of material. So here's one of them from our lab. That's a 5-cent coin. There's quite a small thing. That one has 98 electrodes on it. This is from Riley. Sorry, I didn't put Riley down at the bottom, but she's done some of the cytotoxicity of the electrode arrays, and basically they're fine. So we have some laser fabrication, microfabrication techniques that we're using. There were some things that we were a little bit worried about on what we would be introducing into the tissue, and Riley's done a very nice analysis of this and has raised our confidence level significantly. So apologies for those of you who are still digesting your lunch, but this is the surgical implantation. The limbus, which is the transition between the white part and the iris of your eye, or the cornea of your eye, is up there in the upper left corner. We go back about 7 millimeters, make a little incision, and we slide this electrode array around the back of the eye to the central vision, the area of central vision. And then we can deliver electrical stimulation impulses and record those on the visual cortex and hope that they look like vision. This is a device, this is one of our devices that one of our partners in Korea has implanted. This has been in, I think, for 2 years at the moment. This one was taken some time ago, about after a year, and performing very nicely. This particular image highlights why I like this suprachoroidal approach that we're using. There's the sclera, which is the fibrous tissue on the outside of the eye, and then there's the choroid, and the electrode array is placed very nicely between those two surfaces and held very stably and nicely in that location nearby. Because this was a Smart Surfaces conference, I thought I should say something about Smart Surfaces. We're laser modifying platinum surfaces. So on the left-hand side, we have some platinum that just comes from the manufacturer, and then we laser roughen it, and we get some significant increases in the surface area, which is an important thing for injection of charge. So that's it for basically the Smart Surfaces that I could tell you. Okay, so does it work? So we can build all these things and play with gadgets and do all sorts of stuff in our lab, but at the end of the day, we have to show that this thing works. So some friends of ours in Germany have built this thing called an ImTech neural recording probe, and we can build this thing on the right here. It doesn't have the colors on there. I've done that for highlighting. But generally, we have this polydimethylsiloxane plus platinum electrode array. We can put that in the suprachoroidal space, and then we can stimulate with it. We want to know, does it work? So our friends in Germany have put this device together that we can implant into the visual cortex. We can stimulate at the retina, we can record at the visual cortex, and we can see whether or not the signals get back there look like what vision should look like. You can flash a light, you can get a signal, you can stimulate, and you can get another signal. So we're trying to understand this whole situation about the electric fields and what is it about electrical stimulation that makes these retinal ganglion cells fire. That's all stuff that we're still studying and trying to get a handle on it. But for the time being, what we can do is stimulate at the retina, record at the visual cortex, and if we look at that previous array and up at the upper left of the screen there, we stimulate with one set of electrodes in a hexagonal format. We stimulate and we record neural spikes, neural activity at the visual cortex. We get quite a lot of activity on one electrode in this case, and then we shift to another hexagon or another electrode and stimulate that. We get a very different pattern. Now, this is the closest thing that we can get to something called psychophysics. We can record on the visual cortex, and if the animal or the person that has this sort of thing doesn't see something different with these two different bits of activity, I would be very surprised. But we can't actually guarantee that that's going to happen. It would be very surprising if it didn't. So then if we stimulate all or both of the hexagons at the same time, we get another stimulation pattern again. And interestingly, if we add the two left panels together mathematically, we get something very much like the one that we got when we stimulated both electrodes at the same time. So nice separation, the ability to stimulate one spot, get a response, ability to stimulate another spot, and get a different response, which is very important if we're going to form some sort of pattern vision. And we've been this is very preliminary data, so don't take anything away with you on this, but this is much closer together than the previous study, and we're shifting around with different electrode return path configurations, and we get different responses each and every time. So very promising in terms of the ability to have some sort of spatial recognition between electrodes. And I know we're behind time, so I'll stop there. And if you have any questions, I'm happy to answer.
Process to attempt to mimic the auditory signal?
Yes, absolutely. No doubt that there's some difficulties in being able to mimic the actual activity of a retinal ganglion cell, because if it's an off retinal ganglion cell, it will behave in a particular way. If it's an on, it will behave in an entirely different way than that. The best I think we can do with this particular approach is to send the brain some information and have the brain sort of work it out. If we try and mimic individual pulses from individual retinal ganglion cells, that means we have to go back to those first slides that I said there. You have to intervene individually with the
so it's an attempt to teach the brain what a signal might be and interpret that much the same as you the auditory process developed.
Yes, and the same sort of thing happened with cochlear implants. You threw a bunch of information at them, and the people surprisingly made a great deal of sense out of it. And we hope that the same sort of thing.
Can I just ask a question, follow on from Jeff's? In the previous talk that I've heard Minas Coronio give, but the best, being an ophthalmologist, the best that you hope out of this is that you will begin to see, you'll never see a person's loss.
Someone curing blindness. I think economic meltdowns and all that sort of thing, but if you're going to go out of business, that's the way to do it. I think, and I'd like to be put out of business by that way. We've heard promises about stem cell, the wonderful possibilities with stem cells, and I think that they haven't materialized yet. And there are many things that we hope to be able to do with engineering that will make this better and better and better. Trying to take a pragmatic approach. What can we do now? What can we do by 2013 and have people assisted? Mainly these things happen.